Okay. Yeah, thanks, Krazi, and thanks, Daniele, and the other organizers for doing all the massive sort of work to put something like this together, this new format. So the motivation for what I'm speaking about today comes from, work on the basal ganglia. Okay, so I have here just a cartoon of a coronal section through the brain, and you can see various brainstem areas that together constitute the basal ganglia. So we have the globus pallidus, for example, substantia nigra, and basal ganglia gets a lot of attention because it is a major site for the release of dopamine, a major reinforcement chemical. So it's associated with reinforcement learning and reward. And it's hypothesized to play important roles in action selection and decision making. So this is, for example, one of the subcortical structures involved in decision making that was briefly mentioned in Ann Churchland's talk this morning. So in the picture on the right, we have these various light gray regions, which are parts of this distributed basal ganglia complex. And the idea here is that this is a framework we can think about for action selection. So if we start with inputs from the cortex, representing some sort of evidence accumulation for different decision or action possibilities, you can see there are different color-coded pathways through the basal ganglia, right? So in green, we have what's known as the direct pathway. And notice that most of the connections in this network are labeled as inhibitory, right? So once the direct pathway striatal neurons are activated, their job is to try to inhibit the downstream output from the basal ganglia. Some of you will know this as GPI. In rodent, the dominant component here is the substantia nigra pars reticulata, SNR. And so the SNR in general is inhibiting sort of motor pathways outside basal ganglia. So it's trying to prevent premature action, let's say. And so the direct pathway is trying to shut down that inhibition and allow the action to take place. Okay, but meanwhile, it's competing with the other side of the flow here. So we have this excitation coming in from STN, which is trying to keep the SNR activity strong and block actions. Okay, so meanwhile, the SNR is also integrating inhibitory inputs from the GPE, right? So we sort of have this decision maker down at the bottom of the basal ganglia that's having to integrate all of these different input streams, and as a result, adjust its level of inhibitory output, which gates movement in this theory, okay? Great, so if we try to simulate a model of this process, right? So over here in the schematic, it's different color coding and so on. Now CD is the striatal component, and I haven't separated out direct and indirect, so just sort of pretend they're the same population. So we see that the cortical evidence that I mentioned in the previous slide is accumulating, and that's being reflected in the ramping up activity up in the input here in the striatum, okay? And then we have the competition that I mentioned, right? So this striatal input, ramping, is sort of directly projecting to the output down here at SNR and trying to shut it off so that we can act. But meanwhile, the GPE, right, which is a source of inhibition to SNR, this is ramping down because it's also getting inhibited by the striatal input, right? That's this pathway here. So we have a loss of inhibition on the one side, a gain of inhibition on the other side. That's this red pathway, okay? And so those are sort of balancing, plus the loss of inhibition is complemented by a gain of excitation from the STN. So altogether, we have growing inhibition, but also decaying inhibition and growing excitation, all being integrated at the basal ganglia output. And we sort of have this balance, and we can't act until the balance is somehow broken. Okay, so in this model, the way that the balance ends up being broken is eventually the GPE firing gets so low that it can't really get much weaker anymore, whereas the direct pathway activity keeps growing. And this continued growth of inhibition eventually wins. It causes this pause in the SNR. Okay, and just ignore the magenta and cyan, please. The green and the blue represent the two different actions, so left and right. And they're competing, but you can see the evidence here was stronger for left. So eventually the blue part of the SNR, the blue action channel pauses, and we get action, right? So the downstream area, which isn't pictured here, can finally activate. Here in this case, this SC stands for superior colliculus. This is a saccade task, and so we get our action. Okay, so that's all the theory that motivates this project. But there is, as we say in the US, a wrench in the works. Okay, so there's this paper from a few years ago by Fries et al. Showing what happens with some optogenetic experiments done in the basal ganglia. Okay, and this is rodent. And with optogenetics, the experimentalists could stimulate and induce firing in either the direct pathway or the indirect pathway in the basal ganglia. Okay, so remember, the direct pathway is direct. So you stimulate striatum and you expect to inhibit your downstream target. Okay, and they did see that in some of their trials. So we have this collection of trials under inhibition here, all of these rasters showing this nice pause in SNR activity, and you see the mean firing rate dropping, indicated a couple of different ways. However, they also saw that with the same sort of optostimulation, some of the SNR cells responded with excitation. Okay, so here we see the firing rates are going up. Okay, completely counter to what we would think of from an inhibitory pathway. Okay, similarly, from the indirect pathway, we expect a net excitation because we're removing inhibition from GPE and we're causing firing from STN. So we expect excitation. We see that in some of the rasters, but they also recorded plenty of cells where the indirect pathway stimulation caused inhibition down at the SNR. And you can see that in the far right here. Okay, so this is at odds with the idea that these are inhibitory pathways. They are known to be GABAergic pathways, but this got us wondering what's really going on here with the integration of these different input streams, especially because notice a lot of the firing rates here, even in the absence of optogenetic stimulation, are rather high, right? So we have these sort of 15 hertz, 40 hertz, right? In fact, these areas in the basal ganglia, the GP, for example, can be firing just sort of spiking away at many tens of hertz under baseline conditions. Similarly for the SNR. So there's a lot of possible factors that could be changing the efficacy of the synapses in the midst of all of these spikes that would completely change our interpretation of the system. Okay, so the study that I'm telling you about today is brand new. I haven't given this talk before. It's currently in revision, and you can see the title here. Okay, and the results that I'm going to go through to varying extents are listed here on the right. Okay, so we think, according to our modeling, that in fact chloride dynamics are a major factor in these diverse SNR responses that are seen. Okay, we're going to look at how ongoing GPE activity at this high rate can have significant impact on various features of SNR firing, not just firing rate. So I'm going to talk about oscillations and synchrony, and then I'm going to tie this back into the action selection theory near the end. And we've done a lot of different tests of robustness. I will admit these weren't all on our initiative. Some excellent reviewers prompted a lot of these robustness studies, and things here are very robust. I just won't show most of the robustness results. Okay, so in the study, we do this, it's a computational model. It's just 2 compartments, but we do have 2 compartments which is essential for this because the GPE inhibition actually tends to be somatic or at least proximal. So we model it in that way. And then the striatal inhibition to the SNR neuron is much more distal. Okay, so we have a two-compartment model. Okay, and in addition to all the usual sort of Hodgkin-Huxley type equations that I'm not putting on the slide, we have chloride dynamics. We have chloride influx through the synaptic channels, these GABAergic channels. And we also have the KCC2 pump, which regulates potassium and chloride coming in and out of the cell. So the pump is trying to sort of keep pushing the chloride back out to maintain the balance inside the cell. Okay, so you can see here our chloride differential equation, the intracellular chloride concentration. It has the two terms that I just mentioned, the GABAergic term and the KCC2 term. We have to have a conversion factor that I couldn't fit on the slide, but the reason for that is the GABA reversal potential is actually set not just by chloride, but also by carbonate. Okay, and so this conversion factor makes sure that we're sort of adjusting for that and just taking into account the chloride part when we track the chloride concentration. Okay, so that's the setup. We do have, I should say, short-term synaptic depression and facilitation in the model. Okay, but these do not account for the changes that we see in the different SNR responses. Okay, that would be a natural theory, right? Think, okay, well, the synapses start to depress, that can change everything, they can facilitate, but that doesn't account for the effect. Okay, so the Gittes lab, so our collaborators at CMU, did their own optogenetic experiments, and you can see some of those summarized on the left. Okay, so we have here, it was optogenetic stimulation of the actual terminals of the GPE neurons in the vicinity of SNR. So we're making sure that they're activating the GPE terminals that release GABA to the SNR, and you can see this diversity of responses, right? So these are just examples on the top, and then the summary data is in the bar chart down below. So you can see, for example, a very small number of complete inhibition events, like the top one in the panel A1. Lots of partial inhibition, so those could be slowing down without any loss of spiking, or it could be a sustained loss of spiking, which gradually sort of makes way to some spiking. And then we also have excitation effects and a few biphasic effects, where we transition from reduced firing rate to actually increased firing rate. Okay, so this is an extremely diverse set of responses for a pathway that is just always thought of as inhibitory. Right? You stimulate the presynaptic neuron, you have GABA release, postsynaptically you get this whole hodgepodge of things. So that's quite interesting. Now in the model, we looked at a little two-parameter study, right? And we look at how, as we vary the strength of the pump and the GABA conductance, we can explore this space where here we have color coding by the reversal potential of GABA that results. And so we can get all the same different types of responses by sort of varying where we are in this space. So for example, if we make the pump really strong, right, and that can be enough to maintain the chloride balance, and we maintain the complete inhibition that you see in panel B here. Whoops, I think I just advanced my slide. Okay, GPE, right. Okay, on the other hand, if we reduce the pump strength somewhat, but turn up the GABA, the sort of overall chloride tone, we can balance the effects and get no effect. Or if we go even more extremely in that direction, we're actually switching the GABA reversal potential up to -50. You can see that by the red curve over here. And that's enough to switch the response to excitation. We can also get the partial inhibition types. Okay, and you can see in this partial inhibition, the time scale is long enough that we do get this accumulation of chloride, which gives us a gradual increase in E-GABA. And that gradual increase makes E-GABA less effective at hyperpolarizing the cell, so we get this partial inhibition. Okay, so we get similar effects, sorry about that, with striatal stimulation. This is one of those spam calls. Um, so with striatal stimulation on the experimental side, things look similar, but you can see there are far more biphasic responses, right? So we start off with inhibition, and then we get a switch over to excitation. Okay, and sure enough, we never really saw that in the model with our GPE inputs, which were coming into the soma, but when we have the inputs coming into the dendrite, and which has a different volume and so on, then we can see it's easier for the reversal potential of GABA to change significantly, and so that tends to bias us towards partial inhibition or even biphasic responses like you see here in F. Okay, so just looking at that sort of exploration of parameter space, we can at least reproduce the experimental results from various frequencies of optogenetic stimulation. Okay, so now let's go on and think about what this will actually do to the SNR dynamics. Okay, so to do this, we're going to look at PRCs. Okay, and when E-GABA is hyperpolarized, so here we have an example with E-GABA at -60, the SNR membrane potential throughout a spike, so here we have membrane potential in blue, always sits above E-GABA, so E-GABA is having this hyperpolarizing effect. And so that will end up delaying the next action potential. If a stimulus comes in between spikes, the spike that would have occurred at this gray time is now delayed to the blue time. Okay, similarly, if we turn E-GABA up, which we now know that we can do through the chloride, the naturally occurring chloride dynamics, the same stimulus timing can lead to a spike advance, because now the E-GABA is depolarized relative to the membrane potential at that timing. Okay, so we don't have any fancy sort of nonlinearities here that cause sort of counterintuitive advance or delay. All of the switching here is due to E-GABA. So if by hand, right, we were to just push E-GABA up and down, we can look at how stimuli at different phases would encounter different relationships of E-GABA to the membrane potential, and therefore determine whether they'd be depolarizing or hyperpolarizing. And we can summarize all of that here in the lower right. Right, so we have our different advance delay sort of phase response curves based on different choices of E-GABA. Okay, now Jiang and Gutkin looked at this effect already in a paper a while ago doing weak coupling analysis. So here we are not in the weak coupling regime. We're just, for this moment, just drawing these PRCs based on the effect that a full strength stimulus would have. And here are the numerical versions of these, right? So again, I have the same sort of diagram where we have the range of possible PRCs depending on E-GABA in light gray, okay? In each individual panel, 1, 2, or 3, we've picked out a specific E-GABA, fixed that, drawn that PRC in blue, and we can see where the fixed point is predicted to be. And then we can run ongoing simulations, and here we just have a feed-forward circuit, and we can see where the phases end up distributing, and they're all along the PRC, okay? And you'll see in the next slide that, in fact, the phases will accumulate at the fixed point when it is stable, which is what's indicated here in green. Okay, so again, just to, when we tune E-GABA, we move the position of the fixed point, which determines the phase relationship between these two cells, okay, and we change its stability. Now in a bidirectional pair of neurons, they're both stimulating each other. The PRCs change with E-GABA, that's what's shown in blue and red here, but the fixed point location pretty much stays put, and it remains stable. So summarizing all of that, and I'm sorry, I'm realizing there's sort of a lot of information here on some of these slides, but what we've done here in each of these two very simple circuits is just plot the phase difference between the two cells in ongoing runs of the system, just letting things run and letting them kick each other with these GABAergic transmitter releases. Okay, you can see that when E-GABA is sufficiently hyperpolarized, we have a phase slipping effect, right? So usually the phase difference between these two neurons is sort of up here close to 1 or equivalently 0. So most of the time they're close to phase locked, but every so often the phase will slip, and that's what's also shown over here. And you can see in this case, 13 successive spike pairs between the two neurons, you get this gradual transition in the timing of the two neurons relative to each other until they sync back up again. Okay, and as we depolarize E-GABA, eventually we stabilize a fixed point and we lock the two neurons, and you can see the position of the locking changes with E-GABA. With the two-cell network, it's not nearly as interesting. They're just pretty much always locking at anti-phase. Okay, I'm just checking the time. Okay, so let me move on. So basically, we can vary things. This is still on the feed-forward network now. So we can vary things so that the two cells have different firing rates, and we can again get phase advance or phase slip types of relationships, or phase locking, depending on how we tune E-GABA. Okay, so I'm just going to go ahead and breeze past that. Perhaps most interestingly, we then simulate a full SNR network. So 100 SNR neurons, okay, not the size of the full SNR, but a lot more than 2. Okay, we followed information on the sparse connectivity. So SNR neurons are connected to each other, which I should say is what I'm showing here. This is within SNR, and it's GPE that's setting E-GABA. Okay, so I'm no longer looking at GPE's inhibition of SNR. Okay, so within the network, we have this GPE, which is going to set E-GABA. And then we have connections between the SNR neurons, which are fairly sparse. And again, as we tune E-GABA, we can see radical changes in the variability of SNR firing and what these rasters look like. And we can plot spectrograms over low frequency ranges and see that E-GABA drastically tunes the oscillatory power in the network. So we can be in a sort of phase slipping mode or a phase advancing mode at the two extremes, and in between we don't have oscillations. Okay, and so this is exciting because oscillations in the basal ganglia are associated with a lot of different disorders. This is not the range of frequencies that people usually think of. Right? Beta is sort of the superstar frequency range right now, which is more like 15 to 30 or maybe down to 8 to 30 hertz. This is 0 to 4, but we have a brand new paper also from the Gittes lab showing that in these animals, where all this data is from, in the mouse, there isn't really beta. Other people have shown that as well, and we do find significant low frequency oscillations in the SNR under dopamine depletion. Okay, and that's what this data shows, a couple of examples of what the power spectra look like. And here the total fraction of units with various forms of dopamine depletion showing the low frequency oscillations, and none of them are showing beta. Okay, and these persist with M1 lesions. So these are really coming from the basal ganglia. Okay, so basically what I've shown you is tuning the ongoing activity from GPE could adjust E-GABA in such a way that it changes the whether SNR ends up producing oscillations or not. Okay, there's a prediction associated with that, which is if we stimulate GPE under dopamine depletion, we should be able to turn these oscillations off. And we did that, okay, but I'm out of time, so I'm just going to show you the answer here is yes, okay, that prediction works. And finally, coming back to the action selection, okay, the idea in the action selection scenario, remember, is that this lower frequency striatal activity is working to pause the SNR, right? It has to be this inhibitory input that can pause the SNR and allow us to act. And now if we have the GPE sort of setting the E-GABA, we can change the effectiveness of this inhibition. So we can change its ability to pause the SNR. So we could delay our action selection by tuning the GPE. Right? So as we vary the sort of chloride load in the soma or the dendrite, we change E-GABA. Here's two examples here where we see with different thresholds needed to initiate action, we get different timings of crossings. And you can see the results on the lower right, although I'm flying now. The time to act goes up with the overall sort of GABA conductance and chloride load. Okay, so the summary is here. We find in our computational modeling that changes in chloride load can alter these GABA effects on functional timescales. We think that this could be a mechanism, so a sort of different interpretation of what the indirect pathway is doing, where it could tune synchrony oscillations and a sort of threshold for decision making. And as I mentioned earlier, the effects are robust to a whole lot of different things that I didn't talk about today. So thanks for your attention. Sorry to accelerate throughout the talk. But
thank you very much, John. Really, really interesting. And very, very clear talk. Thank you. Are there any questions? If not, I can ask you a question because it is very thought-provoking, actually. This pause in the action, the timing to action, and this indirect and direct pathways, do you think this might be involved in freezing of gait in Parkinson's?
Yeah, so yes, I think that's a great point, and I'm tentatively, like hesitantly, but in agreement, right? I don't jump to any big conclusions, but it really makes sense, right? If we need, at least for certain movements, this pausing mechanism, and dopamine depletion has caused GPE firing rate to change, it could be changing the chloride load, which could make it much harder to do this pause.
Yeah,
so, yeah, spot on.
Yeah, because there has been this hypothesis that this indirect pathway is involved in freezing of gait specifically in
Right.
Parkinson's, but nobody has come up with any mechanism. So when I was listening to your talk, it was really kind of, so very, very interesting. Somebody, Taylor Cow says, looking forward to the paper. So hopefully it's going to come soon. I will definitely be reading it. Any other questions?
I hope that as well.
Yeah. So we have a question here. Would you expect the results to change if you could scale your model to full size?
Okay, so first let me say no, because 100 neurons was a lot more than 2, and we did have sort of the right degree of connections within that model. So I think at least qualitatively, I'd like to believe, that we'd see something similar. Since the door is open, I'll mention we used sort of the mouse firing rates, which are lower than primate. But one of the things we did test for heterogeneity is if we jacked up the firing rates. So now we have 30 hertz instead of 10 hertz at baseline. We do still get oscillations. The frequencies can sort of switch and become a little higher. So, yeah.
Yeah. We have another question from Victor. Do you think there could be a simple pharmacological way to verify the role of chloride channels on the dynamics of I think it's GABA regulation? Yeah.
Yeah. So simple, maybe not, but reasonable, yes. And so now this is on the to-do list for the Gittes lab, but with the shutdown and everything, the timetable for that is probably not very rapid.
One more question by Dmitry Todorov, that last one. In general, do you think in actual disease, this mechanism is robust enough to account for oscillations despite high variability in patients?
Yeah, so that's, of course, a key question. I think this is probably a factor. I'm not going to say this is the source of oscillations, and I'm not even going to say, like there's one frequency band, but people do see delta oscillations in humans, and they've been overlooked until now in favor of beta. But this whole collection of work says to us, hey, maybe a little more attention should be paid to delta oscillations as well.
Okay, so